Hi, my name is Dr.
Gaukhar Baidildinova.
This video is a part of my lectures on evidence-based medicine and epidemiology. Today, we will discuss experimental studies, also known as interventional studies. The key difference between experimental and observational studies is straightforward. In observational studies, researchers observe subjects without influencing them, while in experimental studies, researchers actively expose subjects to an intervention. The most common example of an experimental study is testing a medication. However, interventions are not limited to drugs. They can include various therapies and treatments, such as herbal medicine, diet, physical activity, massage, aromatherapy, occupational therapy, and sound therapy. Experimental studies can also assess the effects of surgical interventions and other medical procedures. Experimental studies are categorized into controlled and uncontrolled studies depending on whether a control group is included. Controlled trials, in turn, can be randomized, where participants are assigned to groups randomly, or non-randomized, where group assignment is not random. I hope you recall the graph from the previous lecture, which outlines the hierarchy of research types. The sequence begins with theoretical research, followed by applied research, and then clinical research, which can be either observational or experimental. When discussing experimental clinical trials, we refer to different phases, 4 in total. However, before a drug reaches human testing, it must first undergo a preclinical stage, meaning first experiments in tubes on cells and then in vivo studies, experiments on lab animals. By the way, the guidelines for describing in vivo studies is called ARRIVE, and you can find it together with other guidelines on equatornetwork.org website. You can see this progression more clearly in the pyramid diagram. Drug research begins with computer modeling, followed by studies on cells, then animal testing. These preclinical steps always occur before clinical trials. Let's take a closer look at these phases. Phase 1 lasts approximately 1 to 6 months and involves 10 to 50 healthy volunteers. Purpose: to evaluate safety and determine the appropriate dosage. At this stage, researchers seek to answer key questions. Is the drug safe? What are its side effects? Additionally, this phase assesses pharmacokinetics and pharmacodynamics, two terms that are often confused. Pharmacodynamics studies how the drug affects the body, including its mechanism of action and the required dose for a therapeutic effect. Pharmacokinetics examines how the body processes the drug, how it is absorbed, metabolized, distributed, and ultimately eliminated. The first phase is the most dangerous one. In 2006, the experimental drug TGN-1412 was administered to 6 young individuals in the UK as part of a Phase 1 clinical trial aimed at treating various types of blood cancers, rheumatoid arthritis, and immune system disorders. Shortly after receiving the drug, all 6 participants rapidly deteriorated, experiencing severe fever and excruciating pain throughout their bodies. Distressingly, their fingers and toes turned black and began to necrose, resulting in one participant requiring amputation. The incident left all 6 individuals in serious condition, prompting an intensive care unit response. A decade later, in 2016, another Phase 1 trial involving the experimental anesthetic BIA 10-2474 tragically resulted in similarly catastrophic outcomes. One volunteer was declared brain dead, and 6 others were left in critical condition due to unforeseen and severe neurological effects. These incidents underscore the critical importance of rigorous safety protocols and thorough preclinical testing in drug development. The tragic outcomes of these Phase 1 trials have significantly influenced regulatory practices worldwide, emphasizing the need for stringent monitoring and comprehensive risk assessment to prevent such devastating consequences in future medical research endeavors. The migration of clinical research to developing countries has become a growing trend in the pharmaceutical industry, driven by lower costs and easier patient recruitment. However, this shift raises ethical concerns regarding participant safety, informed consent, and regulatory oversight. In 2015, The Atlantic Daily published an investigative report titled "The Life of a Professional Guinea Pig," shedding light on the experiences of individuals in the United States who participate in multiple clinical trials for financial compensation. The article exposed the risks associated with such practices, including inadequate long-term monitoring and potential exploitation of vulnerable populations. About 70% of drugs successfully pass Phase 1 and move on to Phase 2. Phase 2. Duration: 3 to 18 months. Participants: 10 to 50 individuals with the target disease or condition. Purpose: to assess efficacy and monitor side effects. Key questions addressed in this phase: is the drug safe? Is it effective? How often and for how long should it be taken? Only about 1/3 of drugs progress beyond this stage. Phase 3. Duration: 1 to 2.5 years. Participants: a larger patient population. Purpose: to confirm efficacy, monitor adverse effects, and assess the risk-benefit ratio. At this stage, safety remains a primary concern, alongside efficacy. Researchers also evaluate whether the drug may be more beneficial or harmful for certain populations. Approximately 25% of drugs receive approval at this phase. Phase 4. Once a drug is approved for public use, Phase 4, trials continue to monitor its long-term efficacy and safety in the general population. As more data becomes available, this phase may also explore the drug's effectiveness in different populations or conditions, potential new indications or combinations with other treatments. As you can see, all research data must be registered with local regulatory agencies such as the FDA, USA, EMA, Europe, and others. We will discuss these agencies in more detail later in the next videos. A medication is approved only if its benefits outweigh the risks. Out of all drugs that successfully pass preclinical testing, only 5 to 6% ultimately reach the market, after approximately 10 years of development and millions of dollars in investment. Even then, a drug may still be withdrawn from the market if post-market surveillance reveals severe side effects.